So we had question number one of the COVID video project. It's like a general outline of what we will be covering. We're going to talk about the different classes of coronaviruses, including where MERS and SARS fit into the coronavirus order. We're going to talk about the 4 different genera of coronaviruses and their genetic and biochemical differences. And lastly, we're going to talk about the novelty of COVID-19 and how it is different from other coronaviruses that exist today. So coronaviruses are classified under the order of nidovirales, the suborder of Coronavirinae and the family of Coronaviridae. And there are 4 further genera of coronaviruses, being alpha coronaviruses, beta coronaviruses, gamma coronaviruses, and delta coronaviruses. And you can see the 4 different genera right there, being alpha, beta, gamma, and delta. Well, it's commonly referred to as SARS, but it's known as severe acute respiratory syndrome. And it's a viral respiratory illness that originated in China in 2003 and is thought to have originated in bats. And it is classified under the beta coronaviruses genre, group B. And next we have MERS, and it is the Middle Eastern Respiratory Syndrome. And it is also a viral respiratory illness that originated in the Middle Eastern region. And although it is thought to have originated in bats, it was transmitted to camels before being transmitted to humans. And MERS is classified under the beta coronavirus genre, group C. So this image shows the relationship between SARS-CoV-1, CoV-2, and MERS. As can be seen, that all 3 of the coronaviruses are derived from the same natural host, being bats right here. And that's before they were transmitted to humans. And then as also can be seen from this column right here, transmission does not directly occur between bats and humans in all cases, as SARS-CoV-1 passed through cats and MERS-CoV-2 passed through camels. Currently, it is unknown if COVID-19 was transmitted through an intermediate host before reaching humans.
So we know that the spike protein binds to a receptor in order to enter and infect cells. In coronaviruses, 3 spike proteins specifically assemble into what is known as trimers, as seen in this photo by the red, blue, and green. All of these trimers on the surface give rise to the corona or crown-like structure associated with coronaviruses. Beta and gamma coronaviruses specifically cleave their spike proteins in half, while alpha coronaviruses do not. More differences between the 4 genera can be seen in the spike protein and the different receptors used as highlighted in this table. Based on known coronavirus receptors, many alpha coronaviruses utilize aminopeptidase N, or APN, while there's more of a variety seen in beta coronaviruses. Here we can see some variations in the mechanisms used to gain entry to the cell and the different receptors. Both SARS-CoV and SARS-CoV-2 use ACE2 as their receptor in combination with TMPRSS2 as their cofactor. However, MERS-CoV uses a different receptor and binds to dipeptidyl peptidase 4, or DPP4. Coronaviruses come from incredibly large genomes of RNA viruses that can contain up to 30 kilobase pairs. To understand the magnitude of coronaviruses, the human genome has just around 6.4 million kilobase pairs. In this diagram, we can see the 4 main structural proteins of coronaviruses: the spike, the membrane, the envelope, and the nucleocapsid.
So as previously stated, coronaviruses have great diversity. There are 4 genres of coronaviruses, with each having slight differences in genetic and biochemical makeup. So the alpha coronaviruses, which are shown right here, they are derived from the bat gene pool and are known to infect mammals. And in humans, the most common alpha coronavirus that is seen is known as the common cold, and it's named HCoV-NL63. So next we have the beta coronaviruses, which is where you find SARS and MERS. And they are derived from the bat gene pool and are known to commonly infect mammals. So once again, those are right there. And then next we have the gamma coronaviruses, which are right here. And they are known to be derived from the avian and pig gene pool and are known to infect avian species. And lastly, we have the delta coronaviruses right here, which are derived from the avian and the pig gene pool and are known to infect avian species and some mammals, but not humans.
If we take a look at the last subset of the beta coronaviruses under MHV, we can see a component that says HE. A key difference is the presence of a fifth structural protein, hemagglutinin esterase, or known as HE, as seen right there, which binds sialic acids on glycoproteins. High levels of sialic acids are present on all mucous membranes, creating high density receptor environments that allow more rapid binding. This is why coronaviruses are primarily transmitted through respiratory mucus droplets by means of a cough or sneeze, for example. While general coronaviruses are not new, there are a few key differences and features of SARS-CoV-2 that have never been seen before, making it a true novel virus. First, SARS-CoV-2 has a much higher human ACE2 binding affinity than SARS-CoV, promoting increased efficacy of cell entry. This is why so many potential treatments so far have been targeted at weakening that bond. Interestingly enough, the overall HACE2 binding affinity of the entire SARS-CoV-2 spike is comparable to or less than SARS-CoV. This means that it has been less exposed, supporting the novelty of COVID-19. Unlike SARS-CoV, the spike SARS-CoV-2 is pre-activated by furin, creating a reduced dependency on target cell proteases, which promotes enhanced entry. Furin specifically enhances the viral entry of SARS-CoV-2 into different types of HACE2 expressing cell lines, usually seen in lung epithelial and lung fibroblast lines, which is why, again, we would see such a prevalence of respiratory issues associated with SARS-CoV-2. In this diagram here, you can see these down and up conformations that either hide or expose the receptor binding domain. These conformational changes promote a hidden receptor binding domain that is able to come and sneak in for ultimate entry. These key differences and features that SARS-CoV-2 exhibits not only justifies that it is indeed a novel virus, it reasonably allows SARS-CoV-2 to maintain efficient cell entry while evading immune surveillance and ultimately contribute to the widespread of the disease. Here are our citations, and we hope you enjoyed our presentation.